My name is Maya Johnson, and our project, overseen by Dr. Michael Mintz of the T. Roche Lab, investigated cellular heterogeneity in viral-driven cancers. Specifically, we focus on the influence of the Epstein-Barr virus in nasopharyngeal carcinoma. Our objectives were to find out how the expression of EBV genes differs between malignant and benign cells, and how the tumor microenvironment composition changes in relation to EBV. Let's unpack that. Epstein-Barr virus, also known as EBV, is a highly infectious herpes virus prevalent worldwide with between 90 and 95% of the global population being infected. Whilst most cases remain asymptomatic, occasionally the infection can cause diseases such as mononucleosis, or mono. The virus infects B lymphocytes and epithelial cells and is able to switch between a lytic and latent state to evade detection of immune cells and produce large numbers of virions to infect other B lymphocytes within the host. This allows for persistent infection. It's the viral DNA and proteins within infected cells that disturb normal gene and protein expression by upregulating oncogenes that stimulate cell growth and proliferation. This in turn increases the likelihood of malignancy, linking EBV to 1% of global cancers. This includes stomach cancer, Burkitt's lymphoma, and the cancer we analyzed in our research, nasopharyngeal carcinoma.
Nasopharyngeal carcinoma, or NPC, is an aggressive form of head and neck cancer associated with EBV that occurs mainly in the epithelial cells of the nasopharynx. This tumour variant is found predominantly in southern Asia, the Arctic, and northern and eastern Africa. For our project, we reanalysed a single-cell RNA sequencing dataset of 15 patients provided by Chen, UP et al. Importantly, while characterising cell diversity in NPC, this study did not investigate EBV on the single-cell level. This data was collected using the 10x system. 10x single-cell RNA sequencing works as follows. First, every cell is attached to a bead with a unique barcode. This bead captures mRNA poly A tails. After several steps of purification, amplification, and sequencing, all mRNA molecules from a single cell will have the same unique barcode, allowing us to compare mRNA expression between single cells. Sometimes, 2 or more cells are captured on one bead, resulting in what are called doublets, which must be found and removed prior to the data analysis to prevent skewed data, as they can wrongly appear to look like intermediate cell types. The dataset we created was conformed by a large matrix within the specific cell barcodes as column names and the genes expressed as row names. Each cell had an entry if a specific gene was expressed within the transcriptome. In order to analyse the data, we added custom references and the EBV transcriptome, realigned the FASTQ file of our dataset to our custom references, reannotated the cells, and defined cancer cells as epithelial cells whom expressed a significantly different transcriptome than the endothelial cells, fibroblasts, dendritic cells, and macrophages. To visualise our data, we mainly use UMAP, a method that projects high-dimensional data onto a weighted graph, with edge rates representing the likelihood that 2 points are connected.
Now to our results. Our first approach was to label the cells by patient and cell type. By looking at this UMAP, we can see that non-epithelial cells tend to cluster together, while epithelial cells, which are most likely cancer cells, form separate clusters. This is expected, as cancer cells have a unique patient-specific mutations. This means that comparison across all patients is not advisable, because each patient's cells express highly different genes, as they are different individuals, and each patient has a different number of sequenced cells. A direct comparison across patients would draw our attention to insignificant genes and/or skew our data in favor of the patients with more cells. We then proceeded with the plot labeled by cell type. From this plot, we could derive that doublets are adequately removed and that epithelial cells are divided into 2 groups. Additionally, we labeled the cells by their EBV expression. This is shown in this plot. We can see that EBV is predominantly found in epithelial cells. Now to our results of the CNA analysis. We were able to identify 3 different subclones within the tumor of patient 1. These subclones are shown in this CNA plot of the whole transcriptome of the patient. The y-axis of the plot shows the cells of patient 1. The x-axis, on the other hand, displays the whole human genome, organized by chromosomes. Each epithelial cell of patient 1 has one row where the color blue indicates that a chromosome is deleted, while the color red indicates that a chromosome is amplified. Hence, we can, for example, see chromosome 8 is deleted in all subclones, whereas only subclone B has the first arm of chromosome 2 amplified. Subclone A can be differentiated from subclone B and C by an amplification of chromosome 9.
To better understand the role of EBV heterogeneity in cancer cells, we first identified cancer cells by selecting epithelial cells with chromosomal aberrations. Next, we labeled these cells by patient and by EBV positive or EBV negative status. This allowed us to not only see the variety in EBV expression across patients, but also analyze the differentially expressed genes between EBV positive and EBV negative cells in every patient. Thus, we were able to see how EBV altered the genes expressed by cancer cells.
In contrast to the diagnosis of EBV infections that were made in the clinic, the results from our single-cell analysis showcased a distinction within patients 3 and 10 who tested negative during the blood test, but expressed EBV genome within their malignant and benign cells. Contrariwise, patient 15 was diagnosed with an EBV infection but had no significant expression of EBV genes within his TME.
We also wanted to study the role of EBV in normal, or non-malignant, epithelial cells. We define these as all epithelial cells lacking chromosomal aberrations, and filtered out any patient who did not have a significant number, greater than or equal to 10, of both EBV positive and EBV negative normal cells. Next, we analyzed each patient's differentially expressed genes across their EBV positive and EBV negative cells. Just like with the cancer cells, we were able to see how the presence of EBV altered the genes expressed by normal epithelial cells. This indicates a premalignant signature of epithelial cells infected with EBV that have not yet become oncogenic. Finally, we analyzed the patient's tumor microenvironments, or TMEs, and found 2 different patient groups based on EBV response. One had lower EBV expressions and higher percentages of macrophages and NK cells. Meanwhile, the others had higher EBV expressions and more B and T cells. This is particularly interesting because B and T cells help make up the adaptive immune system, while macrophages are part of the innate immune system.
Applying single-cell analysis increases the precision during the identification stage of EBV genes. This technique could furthermore lead to more adequate treatments on a case-by-case basis, given that the difference in microenvironment relates to the effectiveness of therapies.